I'd like to welcome for this afternoon section. As I mentioned here previously, the format will be the same as the section in the morning. The topic here is porous materials, porous media applied to smart materials. We are going to have 4 speakers. And I will be chairing the second and the fourth speaker, and I'll pass the word to Professor Siddhartha Lima from the Federal University of Rio Grande do Norte, who is going to chair the first and the third speakers. So Siddhartha, I'll pass the word to introduce Professor Rosângela. Thank you.
Okay, Márcio, thank you. So let's go begin the section of porous media and smart material. I have a great pleasure of introduce the talk of the Professor Rosângela Balaban from Federal University of Rio Grande do Norte. The topic is porous and smart materials made of polysaccharides. Dear professor, you have 30 minutes. The microphone is open for you.
Good afternoon. At first, I'd like to thank the organizing committee for the invitation to present this lecture. So, my name is Rosângela Balaban. I'm from Federal University of Rio Grande do Norte. I'm a professor at the Institute of Chemistry. And my talk today is about porous and smart materials made of polysaccharides. So, our institution is located at Natal, that has about 819,000 people. And our university has more than 43,000 students. In our laboratory, we have been working on polymers applied to enhanced oil recovery, water and oil-based draining fluids, scale inhibitors in aqua systems, and porous and smart materials for different purposes. So I will show you some recent results about porous hydrogels. So in this slide, we are showing the application of guar gum in brine clarification and oil water treatment. Guar gum is a polysaccharide extracted from seeds of leguminous. So this polysaccharide was cross-linked through borate ions. And in this picture, in the first picture, we can see oil water of a high salinity that was submitted to guar gum gels. So we added guar gum with borate ions. In first minutes occurred the gelation of guar gum, and along the time, these gels collapsed, trapping the oil. So you had the clarification, the salinity was maintained, but without particles in suspension. And the oil was concentrated inside the hydrogel, guar gum hydrogel. In this second work, we evaluated the effect of thermoresponsive polymer brushes on magnetic chitosan microspheres. So now we worked with chitosan, that's a polysaccharide obtained from chitin, that's an animal polysaccharide, that can be found in the shells of crustaceans. So we used this polysaccharide, chitosan, to obtain microspheres. In this scheme, we have the core made of chitosan, cross-linked chitosan. Inside the core, you have some magnetic particles of iron oxide. And on the surface of the microspheres, we have a thermosensible polymer, synthetic polymer. So these branches are made of poly and isopropyl acrylamide. What happened? Here you have our oil water, petroleum. We always use petroleum as oil phase. So you have our oil water of high salinity. And we added in little b, we added the microspheres of chitosan containing the magnetic particles and finally applied a magnetic field. So it was possible to concentrate the oil inside the microspheres and to separate from the aqueous medium. In the table, in the table, we can see that it was obtained a very high oil removal efficiency using this system. In another work, we prepared eco-friendly hybrid hydrogels made of polyacrylamide and starch. So you prepare different hydrogels with different concentrations of cross-linker, and we obtained very clear hydrogels. And we saturated these hydrogels with silver nitrate. The silver nitrate was reduced to, in the case, the cation of silver was reduced to nanoparticles, silver nanoparticles inside the hydrogels. So here we have a black hydrogels due to the presence of silver nanoparticles. And this system, the hydrogel with silver nanoparticles, were applied in a technique named SEARS. That means, as is here, that means surface enhanced Raman scattering spectroscopy. This spectroscopic technique is very useful. It's useful to improve, to enhance the detection of some organic molecules. In our case, we selected phenol. So, in the picture, we can see the hot spots of silver particles, nanoparticles, and these hot spots can increase the signals of the Raman spectrum. So here you can see very high signals of very high intensity. In the picture, in the graph, we we can see that in the case of pure phenol, we have a spectrum, Raman spectrum, very good, with a high intensity of the peaks. However, when we decrease the concentration of phenol in aqueous medium, the signals disappear. And using this device, it was possible to detect phenol in very low concentrations, such as 10-7 molar concentration. The same hybrid hydrogels we applied to evaluate the remotion of phenol. So we evaluated all the hydrogels, and you observed that depending on the composition of the hydrogels, we can have different behaviors with different capacities of adsorption. The best results were obtained for the first sample with the lower concentration of cross-linker and starch. So it was possible to observe a high capacity of adsorbed phenol inside the hydrogels, and it was possible to deliver the phenol from the same hydrogels. It is very interesting because you can reuse the hydrogel and the phenol delivered in the aqueous medium. In this work, we used starch from mango kernel to produce nanogels. So the starch was cross-linked with glutaraldehyde, and afterwards they were functionalized through carboxymethylation. And poly-N-isopropylamide were attached to the surface of the nanogels through the reaction between the amine group and carboxylic group. In this picture, you can see the nanogels produced, and these nanogels were thermoresponsive at room temperature, about 25 degrees. The branches was opened, and after, with the increase of temperature, the branch collapsed. This behavior can be useful to control the delivery of organic molecules. Finally, I'd like to conclude that polymeric hydrogel networks are suitable materials to prepare devices with appropriate mechanical and chemical properties for several applications. Inside the pores of the polymeric hydrogels, it is possible to trap nanoparticles with different intrinsic properties to obtain, as well as organic molecules, leading to important technological applications, especially in the presence of thermoresponsive moitures. In addition, eco-friendly porous materials can be easily obtained through the use of natural polymers. However, it is necessary to select appropriate routes of chemical modification. I'd like to thank my team of undergraduate, graduate, and postdoc students, our collaborators, and the financial helpers, Petrobras, CNPq, and CAPES. And you for your attention.
Siddhartha?
Márcio?
We still have
You?
15 minutes for the next speaker. Maybe we can just wait, right? Still have 15 minutes. Right, Gregory? Do you have the same time that I have in here?
Yep.
Okay.
Well, I'm not sure who is the next speaker.
Is Elcio
Elcio.
Orlandi? He's the next speaker.
Well, I don't know. As you wish. You are the chair.
Yeah. Okay.
That's the best part.
Okay.
I suggest wait a little bit. Maybe someone is
Yeah.
coming, but we
Yeah,
can take
sure,
advantage of this time to promote some discussion a little bit.
Yeah. Yeah. I agree.
Okay.
So Liz is here already. Yeah. So we are with all speakers.
She's here. She's already in.
Yeah.
I ran
don't
away
see-
from the other one. Hi, Marcio.
okay.
Hi, Gregory.
Yeah.
Okay.
Hi,
Welcome,
Lily.
Lily. Welcome. Okay.
How are you?
I saw your message that you're wrapping up another meeting, right? Oh my God. Very
This is
busy.
supposed to be summer, right?
Supposed to be
Welcome,
summer.
Lily.
How are you guys doing?
We're fine.
Nice.
Surviving.
Seriously.
I think it's better if we wait a little bit until 2:00 for the second talk, and then if anyone would like to ask any questions now, we can ask now and then ask later on too, to have some discussions.
Yeah. Yes, of course. So just to start a discussion, our small familiar Brazilian conference became worldwide meeting. And I make a statistics here for the inscription, but now I just discovered that there were very few options. So actually so people put themselves in somewhere because there were no options for industry or for assistant professors or something like that.
Those people
So-
don't count.
Yeah, no, so this is like projection where people think the closest link they belong. But then you see that we have a quite a number of at least young people from heart, which is nice.
Do you have an option for a postdoc there too?
I'm not sure. We have to ask Siddhartha. I don't remember. Siddhartha, these were only the 5 only options?
You saying this site?
When the people make an registration, they have to click on some options like and I suspect these were only the 5 options. So graduate,
exactly.
master.
Just
okay.
this.
So you see everyone, so put what they are closest to.
Yeah. Exactly.
So are you saying there is some advantages of doing everything virtually? You cover the globe?
Yes.
Yes.
And yeah, but there are some disadvantages. For example, some people can just lost connection.
Hmm.
Which is also possible in presence meeting, but it's hard.
The good thing is that we're not competing with the Olympic Games, yeah, because of the time.
It was exactly the point of this conference. I mean, the people watching Olympic Games, they don't have nothing to do, and then they say, I will see something about porous materials.
Exactly, exactly.
And the conference ends at 4, then you have 4 hours of resting, and then you can come back for I don't know, throwing something heavy.
Yeah, exactly, yeah.
Mhm.
Gregory,
Yes.
so we have time for a quick question for Rosangela, right? Until
Well,
2 o'clock,
you-
right? Here in Brazil,
I'm-
right?
It's Marcio. Marcio is a
Oh,
boss
Marcio.
now.
Yes, Abreu. You can ask. Yeah, since
Oh.
we still have 10 minutes, you
Yes,
can
I'd
ask
like
questions.
I'd like to ask Rosangela to show her presentation again. I have some curiosity
Okay.
about some results, please.
Yeah.
Relate to the experiment, right? Laboratory
Okay.
experiment, right?
Yes. It's okay?
Oh, no, yes, please go ahead.
Oh, sorry. Okay.
Yes. First of all, thanks for your talk. And could you please explain more detail? How is the time consuming of this experiment?
Well, the solubilization of guar is quickly. Quickly you solubilize the polysaccharide. And as I put now this part, this change of this reduction of volume of hydrogels clearly was observed in 12 hours. Okay.
Mm-hmm.
And it was increasing along the time, the contraction of the hydrogel. And it's important, one important point is the pH. The pH of the solution has to be a high alkaline. This changing the hydrogel only works at pH, at alkaline pH.
I see.
I didn't show a lot of details because I thought that it would consume a lot of time. So I prefer to show the principal results.
I see. My point is, I mean, this is to me it seems a very nice result because you are cleaning the situation, right? The water, right?
Yes.
And I would like to know about the scalability for, I mean, an industrial application. We see here in Brazil last year, we have a huge impact of the oil, right? In the bits of the northeast of Brazil, or maybe in some kind of industry. I'm wondering how it could be scalable in terms of industry for the treatment. Could you please mention some word about that? How it is the applicability to the industry?
I consider that it's possible to apply at industrial scale. For example, in big tanks, and but it's necessary. It's necessary. How can I say? A time of without changes, without disturbance. But I suppose that it would be possible to use in industrial scale.
I see.
But it is, yes, I suppose, as the other types of purification of removal of oil are applied.
I see. I see. Maybe some kind of patents, maybe.
Sorry?
I mean, I'm wondering that if you have this kind of application for Petrobras or some industry.
So I consider that it would be necessary a project of adaptations to industrial scale, because this work happens at the labor scale, so it is necessary to some adaptations.
I see. I see. Very interesting. Please, Márcio, go ahead.
Professor Rosângela, do you also work on polymer flooding methods to?
Yes, yes.
And you use these methods to enhance the viscosity of the water. How do you apply your methods in polymer flooding? How do you? Because I saw that polymer flooding is usually used to decrease, the contrast in viscosities, and you can minimize viscous fingering. So this experimental work that you have been doing also, you can study experimentally what's the change in the viscosity of the water. Is it possible? Can you comment on that?
When think about polymer flooding, it is we work on polymers that have been working on polymers that doesn't cross-link, but only are thermal responsive, because inside the reservoir is very important to obtain a high viscosity, polymer viscosity, to enhance the displacement of the oil. So, but in general way, it's recommendable the use of polymers of very high molecular weight to increase the viscosity. But the very high molecular weight is not stable mechanically. That means the molecular weight decreases during the pump. In some works, we produced thermoresponsive polymers with lower molecular weight, but inside the reservoir at the high temperature, the polymers associate themselves, increasing the hydrodynamic volume and the viscosity inside the reservoir, but not precipitate, but not gelified, only aggregates, improving the viscosity.
Okay.
This is the idea.
Okay.
I didn't show here because of the theme of the conference, porous materials. So I chose the porous materials that I have been working.
Okay. Thank you. I think it's 2 o'clock now. We have to move to the next speaker. I'm very happy to. Can you cut your sharing, Professor Rosângela, please?
Okay.
And Professor Elso, can you share your screen? So I'm very happy to introduce the second speaker of this section, Professor Elso Orlandi. He's from the Mechanical Engineering Department of the Federal University of Rio de Janeiro. So welcome, Elcio. The floor is yours.
Oh, thank you very much, Marcio. Can you hear me?
Yes, clearly.
Okay, so thank you very much for the invitation. I also apologize that I could not be here the whole morning because I had to teach. So the title of my talk is Thermal Treatment of Cancer. Okay, so this is a brief summary, the motivation, past and current works that we have been doing, and some conclusions. And this is a broad list of collaborators that somehow their results are in this presentation. So this thermal treatments of cancer, they can be classified as hyperthermal or thermal ablation. This term hyperthermal, it's used when heat is used to make the tumors more vulnerable to other kinds of treatment. It's usually associated with an increase of apoptosis. This is in the range between 41 and 45 degrees centigrade. For thermal ablation usually refers when heat is solely used for the destruction of the cancer tissues, and it's usually for temperatures above 50 centigrades. When the temperature and the time that the tissues are exposed to these high temperatures, it induces a necrosis. Today, we have radiofrequency ablation as a common medical procedure to treat tumors, although this is usually intrusive. So the radiofrequency antennas, they are actually located near of the tumor or even inside the tumor. So the idea with the advancement of nanotechnology, actually the idea of having a non-intrusive heating procedure was possible because nanoparticles, they are usually about they are in the subcellular size, so they can be made to target specific cancer cells and promote drug delivery, although toxicity is still a question. In fact, people have observed that the nanoparticles in the bloodstream, they tend to naturally accumulating in the tumors. Also, nanoparticles, they can be designed and, in terms of their materials, the geometry, and also their dimensions to increase the absorption at some wavelengths. Because especially metallic nanoparticles, they exhibit what is called surface plasmon resonance. So we can see in this figure here, for example, gold nanospheres, they become resonant about this wavelength here. And so our interest has been in the application of inverse products and also inverse design for both hyperthermal and thermal ablation. And why is that? Because usually properties of tissues, they depend on the physiological state of the individual, and also they exhibit a large variability from individual to individual. So one important point is that it's difficult to predict the thermal dose for the treatment. And hyperthermal and thermal ablation, they also, as any other cancer treatment, they require some planning, so numerical simulations, and also there might be some predictive control strategy associated with this treatment. Why? Because as any other treatment, the objective here is to kill the tumor, but cause minimum thermal damage to the health cells, and that's how the use of nanoparticles in the tumor plays a very strong role, because you can concentrate the heat generated from a non-intrusive source on that region of interest. Also, to monitor the treatment, some techniques have been developed, like magnetic resonance or photoacoustic temperature imaging. So they also involve modeling and uncertainties, and all that comes within the scope of the solution of inverse problems, especially when you consider the uncertainties in the models and also uncertainties in the measurements. We can take these into account in the Bayesian framework of statistics. So our works have been along these 5 lines of action. Like first one of them was the indirect use of indirect temperature measurements, the solution of inverse problems for indirect temperature measurements. Also, it became we noticed that it was important at some point the question of model selection and also model calibration, especially for the thermal damage to cells and tissues. And since this is all evolved with nanoparticles, we have a very good collaboration with a friend from the material science department of COPE, Gilson Santos, for the development of nanoparticles. And currently we are working on the modeling and for the coupled hyperthermic chemotherapy treatment, and how to evaluate this coupled treatment, and also for the design and uncertainty for 2 kinds of external heat sources, like laser heating and also high intensity focused ultrasound. So we started these works on indirect temperature measurements by applying Bayesian filters. For example, like with the radio frequency heating, where we have in this case a sphere simulating a tumor inside the muscle, where we have 2 electrodes, and so with the temperature taken inside the tumor, we can estimate the temperature at different points in this whole region. In particular, even at the point where the measurements are taken, we have a better agreement of our estimates obtained in this case with the particle filter than actually the measurements. The same technique we apply to geometry closer to reality. For example, in this case here, we have a tumor in the pancreas. And we could also get a very good agreement between what would be the exact temperatures and the estimated temperatures. These are all computational works, and most of the time here we'll be talking about computational works, except at some points that I'm going to present some experimental results, actually. The same idea has been applied to the treatment of skin cancers by using laser heating. Like in this case, we have a region here simulating the tumor in the dermis. And by using one single temperature measurement, we can predict very well the thermal damage in the region and also the temperature field in the whole region, as is shown in this case for 2 different times. These are the exact temperature fields, and this is the tumor region here, and this is the temperature field that is estimated with this particle filter solution. And so these techniques we applied actually to some experiments for validation by using phantoms made of a plastic PVCP. And the phantom contained the region with nanoparticles, in this case, iron oxide nanoparticles that we have produced ourselves and to simulate the tumor. And the phantom is heated by a laser, and the surface temperature measurements are taken with an infrared camera. And in this figure also we can see some thermocouple wires inside the phantom that were used. Some of the measurements were used for the inverse analysis, and the others were used for validation. And so we can see here one of those measurements that were used in the inverse analysis, and we can get a very good agreement with the estimates. And even at some points, some regions like here, this is the surface of the phantom, where it's exposed to the laser heating. We can estimate, we can get a very good estimate of the temperature as compared to the actual measurements. In this case, those measurements, they were not used in the inverse analysis and only for validation. And those techniques, and it's interesting because those techniques, we can also kind of estimate parameters of the model. So these are different model parameters that were estimated together with the temperature field in the whole region. But those previous examples, we always used some intrusive measurement of the temperature. So we started to work with other physics to allow for non-intrusive temperature measurements, like, for example, the magnetic resonance in a region that contained a tumor. And it's interesting because when we add the model for this other physics, we can even consider that the measurements are taken at some region of interest, in this case around the tumor, on a coarse mesh. And actually we estimate the temperatures in the whole region in a much finer mesh, especially here in this region that contained the tumor also. And so we can see that the estimates are made here in a finer mesh than that was actually used for the simulated measurements. And these are the estimates obtained with the Kalman filter. This would be the exact temperature in the region, and this the estimated temperature increase actually. And with the same idea, we also worked on photoacoustics. Photoacoustics, it results when, for example, laser pulse is incident on the tissue, and when this laser, and it's a very fast laser pulse, so when the energy is absorbed by the tissues and locally, actually, it generates some pressure waves that can be detected with ultrasound transducers. And the conversion of the energy from the laser beam into mechanical energy, it's in the mathematical formulation, this links to a parameter called the Grunessen parameter, which is a function of temperature. So the idea here was to, again, simulations. So we have the laser heating of a tumor for the hyperthermal treatment, and also an additional laser pulse that creates the photoacoustic waves that are detected non-intrusively by some ultrasound transducers. And the idea is to recover this Grunessen parameter in the whole region, which can be associated to the temperature in the region. So these are some results that we obtained with Markov-Chain Monte Carlo method. This is the tumor region where the largest temperatures and the largest temperature gradients take place, so we can get a very good estimate there and also in the rest of the region. And these are results obtained with the Kalman filter, and it's interesting here that in this case we consider a non-uniform distribution of nanoparticles inside the tumor, which generated a non-uniform heat source there, and then a non-uniform temperature, and this could be very well retrieved by the Kalman filter. Okay, as for the question regarding the model selection and calibration of thermal damage, we developed a model for the thermal ablation of tissues with laser heating. And when we compared to some experimental results available in the literature, we could get a very good agreement between our model and the experimental data for the prediction of the crater that is generated by this very concentrated laser heat. On the other hand, when we consider the thermal damage, we can see different colors of the tissue that correspond to different thermal damage, that because the tissues have been exposed to different levels of temperature, and also for different times at these high levels of temperature. The agreement was not very good, actually the results obtained with different thermal damage models, they were quite different. So we applied this ABC, approximate Bayesian computation technique, to simulate with simulated measurements to actually select the model of the thermal damage. For example, in this case, we start with our computations with two competing models. In this case, these the simulated measurements, they were generated with this model one in blue, in green, I'm sorry. And we see that this is a sequential Monte Carlo technique, and eventually only the correct model is actually selected by the algorithm. In the same line of what we estimated with actual experiments using a radiating heater and also the infrared measurements, we estimated the parameters of the thermal damage model for the Arrhenius thermal damage model with next vivo tissue. Okay, about the development of nanoparticles in collaboration with friend there, Gilson Santos, we recently developed those palladium nanocubes and nanostructure palladium Syria particles, and including their hydrates form. So we can see here the palladium nanocube with dimensions about 17 nanometers. And we tested these different nanoparticles in nanofluids heated by laser also. Here's the well, one of the wells in a 96 well plate, and containing the nanofluid, and the other containing distilled water as a reference, as a basis. And we can see very large temperature increases for different concentrations of nanoparticles. So actually, those nanoparticles increase the absorption of the laser energy, and then we investigated further these effects by solving the inverse problem in a natural convection model that considers the laser beam, and this is the nanofluid inside the well where convective effects take place, and what happens actually is this is a direct simulation that we can see the heat source located along the axis of the well, and where the laser is located, and the non-uniform heating induces natural convection currents, and actually result in very different temperatures inside the well. You see, this is the temperature increase, where it's here around 25 centigrades. At the bottom is around 17 centigrades. So although most of the people use mean temperatures for the estimation of those quantities related to those nanofluids, we use the inverse analysis based on this natural convection model to estimate parameters, different parameters of the model. And by using only non-intrusive measurements taken with the infrared camera, which they actually agree very well with the model after the parameters are estimated, as shown by these two figures. Oh, and why that? Because we want to work with the coupled hyperthermal chemotherapy treatment. So far, we have developed the full framework for the Monte Carlo direct and inverse problem simulations for the in vitro experiments, where we have the cell culture containing a chemotherapy drug, and this cell culture is heated during some period, and how this heating and the chemotherapy influences the number of cells in this in vitro experiment. And this is actually a continuation of some work that we already did on only the chemotherapy analysis without the laser heating, which we plan to do in the future. Okay, so finally, for the design under uncertainty, we have worked with more or less the same cases that we did before, and the aim here is to be able in the future to prescribe individual treatment, individual in terms of, for example, in this case where we have the laser heating of a skin tumor, in terms of different parameters, like different design variables, like the laser fluence, the fraction of nanoparticles inside the tumor, and also, for example, even like if we have pulses of this laser, what would be the duration of these pulses? What would be the duration of the treatment? And the idea here was to still work within the Bayesian framework of statistics and consider this design in terms of a probabilistic sense. So, for example, we define what would be equivalent of a likelihood function. We define what would be our desired objective. In this case, for example, it was the thermal damage inside the tumor, and then with the also considering the Metropolis Hastings algorithm of the Markov chain Monte Carlo method, we can get the design variables, and actually would be their posterior distributions that could allow those uncertainties in our objective. And we also have done this for transient, for a laser influence without a fixed functional form, a time variation functional form, right? like for example, before we did for pulses, and here we are considering a general function that would be estimated, and actually they tend to our desired objective very nicely. And same thing was done for the high intensity focused ultrasound, where here we have a illustration of the transducer, of the ultrasound transducer. Here would be the tumor inside the tissue, and this would be, for example, the resulting optimally designed thermal damage inside the tumor. Okay, so finally, to finish my presentation, let me mention that for all those cases, I have considered that somehow the nanoparticles, they are somehow distributed inside the tumor, but an important point of study is how those nanoparticles, they actually are they are distributed inside the tumor, and nanoparticles at the cellular level, the nanoparticles they are usually taken by the cells by endocytosis, and but for the tissue, we have like the blood streams taking the nanoparticles to the tumors, but diffusion plays a very important role here. No? And so those results they represent here, they were obtained with spheroids, and which are cell aggregates aimed at simulating tissues. In this case, it was an ovarian tumor. No? There is no blood flow towards the spheroid. So here, basically, what we have is diffusion. Diffusion from those spheroids, they were inside nanofluids, and so in this top figure, we have the different cuts of the different cut views of the spheroid, which were grown at 37 degrees centigrade. You know? And these are fluorescence measurements that indicated the concentration of the nanoparticles. So at 37 centigrades, most of the nanoparticles, they were located at, near the surface of the spheroid. But at 43 centigrades, the diffusion was enhanced towards the interior of the spheroid, and actually the distribution of the nanoparticles was more uniform inside the spheroid. So this is something that we are starting to work right now. So I'm about to finish some conclusions that we have been using those Bayesian techniques for the solution of inverse problems, and also for design problems. Many of our computational work involve a simple geometries, and we need now to work on more realistic cases involving 3D geometries and multiple organs also. And also we need to work further on with actual experimental measurements, particularly at the cellular and also the cellular scale, and also at the scales of tissues, for example, like spheroids. But we are very happy with those results that we obtained with the development of nanoparticles, especially because they could be hydrogenated, and hydrogen can be used as an antioxidant for the tumors. So this concludes my presentation. I would like to thank you very much, the organizers of this InterPore conference, and especially Márcio Moraes for inviting me. It's an honor for me to be here. I'd like to thank CNPq, CAPES, and FAPEV, and well, I guess that's all. Almost in time. Thank you.
Okay. Thanks very, very much, Elsa. Okay,
Thank you.
so I will pass the word to Siddhartha, who is
Okay,
going
Márcio.
to introduce the third speaker, the next speaker. And the questions will be posed after the fourth talk. Okay, Siddhartha, I'll pass the word to you.
Okay, Márcio, thank you. First of all, we'd like to thank the Professor Rodrigo Weber for accepting our invitation. Professor Rodrigo Weber from Federal University of Juiz de Fora in Minas Gerais State. The microphone is open for you. Thank you, thank you.
Okay, thank you very much. I'll try to share my slides here. Okay, where is it?
Yes, okay.
Can you see it?
We see.
Can you hear me?
Yes, Rodrigo.
Okay, thanks. So I'd like to thank for the invitation. The conference is being really interesting. So I'll talk about modeling perfusion cardiac tissue to support the diagnosis of infarct, COVID-19, and other not so pleasant diseases. And so it was a little bit difficult to fit into the title, but so where I'm from, so we are from Juiz de Fora. It's 600,000 inhabitants. It's cold here because it's 1,000 meters high, but it's very close to, so in the middle of these three main capitals of Southeast in Brazil. So it's close to Rio, São Paulo, and Belo Horizonte, the capitals. Our campus is really nice. If you never visit us, it's a very nice campus. We are like 1,000 professors with around 25,000 students. And I had to use a trick of words here to try to fit more or less my talk. So it will be a lot like about smart materials model as porous media instead of smart materials only. And these are the authors. They're all from here from the Federal University of Juiz de Fora, in particular from the graduate program in computational modeling. And so this is my smart material. Some could claim that this maybe the smartest material in the world, which is the human brain. And here we see like what COVID can do to your brain. This is an image from a 54 years old man with COVID, with multiple lesions in these dots. Can you see my mouse?
Yes.
Okay,
Yes.
thanks.
Yes, we can.
Thanks for the feedback. So this is like these white things are lesions or damage or injuries, or there are many names. I'm gonna call it fibrosis sometimes also. And not only for old people, this is for 1 year old and 13-year-old and 4-year-old kids also with COVID. And so it's known that like around 80% of people that are symptomatic have problems have brain neurological issues due to COVID. And even worse, like that, this is a Nature paper from 10 days ago, highlighting that almost half of the persons that are also asymptomatic or had only mild symptoms also have some kind of neurological problems, like 4 or 5 months after the COVID, the acute phase of COVID, after being infected. And these could be something like loss of memory, or what they call also the fog brain, which is like a confusion together with loss of memory. So loss of cognitive due to COVID. Another very, very smart material is our heart. It's really amazing organ, and this is also a magnetic. So I forgot to say, all of these are magnetic resonance imaging using contrast as to highlight the damage of the tissue. And this is again magnetic resonance imaging from a 26-year-old woman with COVID, with myocarditis, so inflammation, and she had these dead tissue after. This is the left ventricle, this is the right ventricle, and this is just to highlight where was the damage in the tissue. So this is like a very good tool to identify damage in different organs, which is magnetic resonance imaging with contrast. And this is the damage can be, there are different reasons for the damage to show up. So it could be just attacked by the virus, or from another disease by some bacteria, or autoimmune disease. Also, even the immune cells attack your tissue and start to destroy and damage your tissue. Or it can be by infarction, a brain infarction or myocardial infarction in the heart. So you have an obstruction in one of the arteries. So some part of the tissue is not well perfused, so it doesn't receive enough oxygen and nutrients, and it starts to die. The cells starts to die. And so there are multiple reasons for these kind of damages, and they both show up in, so you have different diseases like myocardial infarction or myocarditis, when it's inflammation in the heart, which is also can be due to COVID-19, as I mentioned. So here's the mechanism of these contrasts, or these MRI exam with contrast. So you inject these contrast. I call it here CA, contrast agent, and it has a higher concentration inside the vessels. So here the capillary, where there is this interchange of anything happens in the capillaries. So if you have not so much contrast outside the vessels, the contrast will go to the outside of the vessels, so to the extravascular space. And after some time, so the contrast decreases inside the vessel. It's higher inside in the outside of the vessel, and so it goes back, and it's washed out by our kidneys. What happens is that when we have a damage, so when there is a damaged tissue, we have some fibrosis and extracellular matrix that tries to fill that void. There was a cell there, there's no cell anymore, so there's some proteins here. And what happens is that when you have these damage, the contrast agent, this particular contrast agent that's used for MRI exams, is trapped inside these extracellular matrix, this dead tissue or scar or fibrosis, or many names for the same thing, or the extracellular matrix. So it gets trapped here inside. So that's what we see in those images before, like when it was like bright. It's because it was the contrast was trapped in these by these extracellular space or fibrosis. So this is a very nice exam. It provides very interesting information for the diagnosis, but it lacks something about quantification of the damage. So this is another case for infarction. So we just inject again, this is the heart, left ventricle, right ventricle, and inject the contrast, and the heart brightens up by the contrast. But this part here, when there's a part of the tissue that does not receive the contrast, it's because it's not receiving oxygen or or the blood, or the nutrients or the blood. So it indicates that the fusion is not correct here. So after a while, if everything is washed out like here, it indicates that, oh, okay, so we have a problem with the perfusion, but there's no damage yet. So it's like we say it's the tissue is ischemic, so the tissue is suffering, but still there's still time to save it. And there are a lot of procedures then the doctor will do to try to save this tissue. But sometimes after a while you do the exam or you wait some minutes, you see there was a perfusion problem here, you wait some minutes and then you do the exam, you see that the contrast was trapped in the damage. So you see this bright image here, and then you say, okay, there's a damage in the tissue, and it's not ischemic anymore. The tissue died, and this is fibrosis or injury or lesion or many names for that. So this is a very nice example for many different diseases, which is this exam of magnetic resonance imaging with contrast. But it lacks a quantity because it informs you if there is or not damage in the location, but does not inform you like, okay, what's the percentage of damage in this region? Like it's 50% of the cells are dead, it's 10% of the cells that are dead. This kind of quantification we don't have. And it's very important. I will show you why in a minute. So the goal here is to evaluate models for the perfusion of contrast agents that can quantify the degree of injuries in the heart. So not only to see, so the goal here is to reproduce this exam. So we this phenomenon, so that we can better quantify what's going on and try to quantify the damage. Not only know if there is or not some damage, but also to see what's how bad is the injuries. So I will not focus on the brain. The brain was just the first slide to talk about, to say a few words about smart material. I'll focus on the heart because our group works a lot with cardiac modeling. So electric modeling with arrhythmias, electromechanics, cardiac perfusion, which will be the focus of today's talk. Edema in myocarditis, also the focus. If I can, I would say a few words about that. Modeling, these two modeling the heart as a poroelastic material, and also hemodynamics and other things. A lot of things that are very interesting that happens in our heart, and we have a lot of works, different works with cardiac models in this respect. So this is why we are interested in quantifying the degree of damage in your tissue. This is a patient-specific model we developed. So this patient had 2 infarct regions identified by the magnetic resonance imaging with contrast. So this is the 1 infarct, and here there's the 2nd infarct. And we developed a patient-specific model for this patient with these 2 regions with infarct. And then we generate, also using Monte Carlo, we generate many different models for the same patient because we could not quantify what was how was the damage. Like, was 10% of the cells were dead, or 50%, 70%, 100%. So we just did a lot of Monte Carlo simulations with the different percentage of cells dead in these 2 infarct areas. Sometimes we had a normal heart beating, nothing was wrong, but other times, depending on the damage here, we had the case of arrhythmia, very dangerous arrhythmia called fibrillation or sudden death, that just desynchronize completely your heart, and your heart cannot pump anymore because the electric wave, which should synchronize the mechanical pump of the heart, is completely crazy, desynchronized, turbulent, and It's called also electrical turbulence, this fibrillation. And then your heart is not pumping, and in a few minutes you die. That's all. It's called also sudden death, this kind of arrhythmia. And this kind of phenomena we had a window. We observed that this occurred only in a window which was near the percolation threshold of these density of tissues that were dead. So it's very important to quantify what's the amount of damage I have, not only the size and the location. So with these, we were trying, so we start to develop models based on porous media to try to reproduce this exam of cardiac perfusion with contrast. Porous media is used commonly for, very usually used for tissue modeling, because the tissue here, you see, this is a cardiac tissue, but could be any kind of tissue actually. So the pink, the dark pink is cells, the pale pink are interstitial space. So you can see, like, you could model these like a porous media, like interstitial space would be for instance, where we have the contrast. These particular contrasts we use, that's using in magnetic resonance, is gadolinium base, and they cannot enter the cell. They can only go to the interstitial space, but they can also stay inside the vessels. Here's a vessel, and here's the vessels, and here also the vessels. And then you can also consider these, and there's another kind of porous material like intravascular. The intravascular domain would be a porous material, and extravascular domain another one. So you have different levels of porous, multi-compartment porous material that has been used since 2013 for the heart specifically, and we also have a couple of papers on that. This is a little bit more about cardiac tissue microstructure. Here are the cells. If you remove the cells, so the gadolinium, the contrast, gadolinium base does not go inside the cells. They can either go in the vessels here, or in the interstitial space here, which surrounds the cell. So and this is the amount of vessels is around 8% occupies 8% of the tissue, of the cardiac tissue, and the interstitial space or extracellular space or extracellular matrix here sees 30% of the tissue. So this is we're going to model these as a continuum, as a porous media, and we'll try to propose models for this perfusion of contrast in these media. So this is the model we proposed more recently in this paper. Was like we had this multiple domain model. We have the arterial domain, the capillary domain, and the venous domain. All these in the intravascular domain. In the capillary domain, we have these exchange to the extravascular domain, where the construct can go to extravascular. And in the case we have infarction, the construct will be trapped here in the infarct. So I have these different kinds of domains. And in another paper also we had, where I show here, we have also like when the vessels are very big, we have a discrete, we treat it with discretely. We use a discrete model for the vessels, but for the fine vessels, this we cannot do it. It's computationally really expensive, so we assume a continuum of vessels of these, and we use porous media for these approximation. To simplify, just to show you some first results, we just collapse all these domains to one intravascular domain that could exchange to the extravascular domain the flux, the contrast, And once again, the contrast could be trapped in the infarction or fibrosis or damaged tissue. And we have these recirculation, yeah, because the blood goes out of the heart and comes back again, and the contrast also goes recirculate. So you have these multiple waves passing through your heart that we would like also to simulate, represent. So this is our very simple model. This is one of the a simplification of these models we proposed. So for the intravascular domain, so here omega I is the intravascular domain. We just assume Darcy's law. We just for the flux, for the intravascular flux, and so the contrast inside the intravascular flux is described by these first equation here. So this is the intravascular concentration of the contrast. It's advected. It's diffused, and there is an exchange of mass of contrast to the extravascular space, which is the second equation here in the extravascular space. So mainly there's only diffusion, so there's not much of advection in the extravascular space. So there's mainly diffusion, and in the case of fibrosis, there's another exchange term here that will be transfer of mass to fibrosis and these equation then models that the contrast is trapped in these fibrosis region. And this is a classical model of adsorption for porous media, for porous material. So we use really like classical models for porous media, like in enhanced oil recovery or many other phenomena for modeling these particular phenomena of cardiac perfusion. And this is like some result. This is the pressure. Here's a normal case. It's also only two D slides. This is the left ventricle, epicardial and endocardial. This is the pressure in a normal subject. Here is just drawing here. This is for if I have an obstruction here, a coronary, a big artery, there's a little bit of obstruction. So I don't have so much of a gradient in pressure here. So you're gonna see that there will be a ischemic, we're gonna model ischemia in this occasion. If I have a very big obstruction in the coronary, so there is even a flat pressure gradient here. So there will be really, the cells will die here, and I will model the infarction here and the damage. And this is the, oh, sorry. I thought it was a movie. I think I removed the movie. So this is the first pass. The contrast in the normal just highlights everything. And after a while, you do again the magnetic resonance, and there's no ground contrast anymore. So here's the contrast when you have ischemic situation. So you on the first pass, you see a shadow here. This will be like a black shadow. There's not enough contrast here because our coronary obstruction was here. But fortunately, this was not so bad. So the tissues didn't die, the cells didn't die. So if you wait and do again the exam, MRI, there's no gadolinium-based contrast trapped here. And in the case of infarct, we have a very severe obstruction here. So you already see again some problems in the perfusion. And when you do again the exam, you see the contrast was trapped in this place, which has a damage, fibrosis, infarct. And we could calibrate the model using clinical data, so we could replicate pretty well what was happening. And with clinical data, here's a healthy patient. Here we see the recirculation. The contrast goes high and goes down, and goes high and goes down, because it's recirculation for your body and going to the heart. And this is what we could reproduce with our model in the healthy subject and a healthy experiment. This was the middle, this one with the ischemic subject. We had, okay, the perfusion is not really good in this region. We see this is a little bit dark, but we can see it. And this is when you have another subject, we had infarct. The time is much longer because you have to do it twice. You have to wait around 10 minutes to do the second exam, to wait for the contrast to get trapped inside the damaged tissue. And this is, in yellow is the clinical data, and black is our experiment. So we could, using our models, we could very well reproduce the clinical data, and we think that this kind of models will be very useful for the quantification of this kind of data. These are some recent results we are generating with another PhD students, Evandro. We're just trying to also model the same, but in 3D, perfusion or myocarditis, biomyocarditis like in COVID-19, we have that I showed before. This is like, again, in 3D, the left ventricle, and we see here the contrast being trapped again by a damaged tissue. And this is the most recent results from Evandro, from a patient-specific, again. This is a patient-specific model of the left ventricle. The patient had 3 damaged tissues, damaged regions, and we could recover these with all this recirculation model, the perfusion model. We could recover these clinical exam. So summary, we developed and evaluated models for the perfusion of contrast agents in the heart. The accumulation of contrast in the injured region was modeled as adsorption, as a classical model for porous media. We performed simulation of myocardial infarction and myocarditis, these 2 kinds of disease. And we have a lot of ongoing work coupling these models with other interesting models like the immune response, edema formation also, which is like when you have really, we see the deformation of the porous media, the deformation of the tissue in the brain or in the heart, because you have accumulation of water during the edema formation. And we use the formal porous media for these kind of studies. We have 2 papers here for, there's a virus, and then there is the immune response here, the leukocytes trying to fight this virus. The fluid phase accumulates here and deforms the tissue, because it stretches the tissue because the edema is being, you have an edema formation. So this was modeled with poroelastic model. And we had another one like a patient-specific one, also that we published. Again, we had a patient-specific. This was with Juliano from Campinas and Thais here from the University Hospital of the Federal University of Juiz de Fora. So they had these images with viral myocarditis, and we could generate again the patient-specific model. And this was what they visually could say, oh, this is where the edema is, and this is our simulation showing where the water accumulation was, and we see that there was a really good match with our model and the characterization done by the medical doctors. So that's what I wanted to show you. So thank you very much for your attention.
Thank you again, Professor Rodrigo Weber, Federal University of Juiz de Fora. Again, we'd like to remember that the question can be posed after the next section. And I give the host again to Márcio Muradi.
Okay, Siddhartha, thanks very much. I'm very happy to introduce the last speaker of the section, Professor Lili. I have a problem in pronouncing your last name, Iagarezian. Is this correct? Or or I
It's
mispronounce it.
it's very close. It's the Armenian version of Ilya. It's
Okay.
You can call me Lilith. It's fine.
Okay. Professor Lily.
So I tell Marcio, I tell my graduate students that unless they can spell my name and pronounce it, I'm not going to sign on their dissertation. So they have 5 years.
Okay.
I do have a lot of fun, yes. Especially at the
Okay.
airports, they look at my name and they go, "What?"
Okay. So she's from the University of Cincinnati, and she was also a plenary speaker, one of the invited speakers of the previous InterPore meeting we have in the chapter. So Lily, the floor is yours, and thank you very much for accepting our invitation. Thank you.
Marcio, thank you so much for inviting me. I really wish I could be there in person. I absolutely loved my visit to Brazil, and I'm delighted to see faces and names of friends that I made there. So thank you again for inviting me. I tried to share my screen and put it in presentation mode, and I'm not sure if this is what's showing. Is that what you see?
It is showing now. Yes, it's perfect.
Fantastic, fantastic. All right, so I'd like to talk a little bit about our work on design of materials with target properties and microstructures. And it is a bit of a continuation of the work that I showed 2 years ago, and I'm delighted that I can actually do that. So yes, designing materials with target properties and microstructure. Now, the motivation for this work is actually quite interesting. It is the lack of real-time microbial sensing in water. So we do not have any sensors that can tell us in real time whether there is microbial contamination in surface water, and that includes streams and rivers and lakes. And currently, you have to take a sample of water, take it to the lab and process it. And it will take more than 24 hours before you know if there is any contaminants. And what is really needed is some cheap autonomous mobile sensors that can sense pathogens like E. coli and give us information about them. So what you see here, I'm going to play a short video. It's an artistic rendition of how we envision a sensor like that. And you see that it's swimming and the surface is kind of undulating. It's interesting because a lot of microorganisms actually swim using this mechanism by undulating their surface. And what you see on this slide on the right is the lab prototype of that sensor. We synthesized it using polymer hydrogels. And I'm sure Professor Rosangela is going to be very happy to hear that, because this is just another type of polymer than what she's using. The propulsion mechanism here is volume phase transition, and I'll talk a little bit about it. And this sensor is capable of transporting cargo, so we can put some beads and things on it and it will move with it. It can sense E. coli. But today I want to focus on the materials research that we've done on these sensors, especially on the microstructure. So we look at what's inside and what is the material that the sensor is made of. So the building, the foundation of the material is a hydrogel, and hydrogels are highly porous polymer networks that hold about 95% of water by volume. The one that we're using in this work is called polynisopropyl or PNPAM hydrogel. And that is a smart stimuli responsive material. And it has a very interesting properties, that volume phase transition, that at the critical temperature of 33 degrees, if you go above it, the gel collapses and expels all the water, and the volume is much smaller than what it used to be. And below the critical transition temperature of 33 degrees centigrade, the gel starts absorbing water and it becomes bigger and bigger and bigger.
So we harvest this capability of this material to empower movement. The interesting thing is that the performance, the movement, how quickly this hydrogel will absorb and expel water really is critically dependent on the microstructure of the porous media that comprises this hydrogel. So the image on the left you see is made with a hydrogel with weak crosslinks. And the result of that is that there is very little connectivity within this material and the pores are poorly defined. And the image on the right shows a strong crosslink that was used in this polymer network, and you see very well-defined pores, and this material will have much higher permeability. So it is really critical for us to understand what is the internal microstructure of this material in order to improve their performance in terms of speed and other things. And so doing this experimentally, trying out different crosslinks, different chemistries, and seeing how fast this material can move is a long and costly enterprise. So what we're looking for is numerical design. And what we want is a fast and efficient method that can generate porous structures with target properties, whatever we want. We want a method that can create a wide range of materials. We want a method that is computationally efficient and that has simple and physically relevant input parameters. And one that we have developed is called the Adjustable Level Cut Filtered Poisson Field. It's a long name. It is a method based on Poisson random fields. We use it for numerical design. It is computationally efficient. It does have very intuitive, physically relevant, simple input parameters. And to calculate how, what is the final parameter that we're measuring, be it pore size distribution, porosity, or permeability, we're using a pore topology method called PTM that we also developed in-house. So we're coupling these two methods, the ALCPF that we developed specifically for numerical design of porous materials, plus the pore topology method that we use for material property calculation. So I want to show you just a quick demo of how this coupling of two methods generates materials. Here we want to control three different properties. We want to control porosity, pore size distribution, and permeability. So what you will see is we will create a domain, we will introduce random seeds, we will generate a Poisson field, we will cut it, and then we will iterate until we hit the properties, the target properties that we want. So let's say we want to meet the parameters of target 50% porosity. We create a domain, we throw in some random seeds. This is our Laplace Poisson field that is generated. We're going to cut it. And we have the final design with the target porosity of 50%. Now the next one is, let's say we want to hit a particular pore size distribution of 5 microns with a standard deviation of 1. Again, we go through the same procedure. We generate the domain, we throw in the seeds, we cut them, we iterate by computing material properties several times until we hit the target. And the final one is permeability, and this is also achieved very quickly. I believe there was under 10 iterations. So this is a very fast method. Both of them are fast. The one that generates the domain and the one that computes material properties. Now, what you show, or what I just showed, it was a homogeneous material. There is no gradient, there is no change of property in any particular dimension. And what you see here are a set of very simple input parameters. Lambda is the seed density, is when we create that empty box and we throw in a bunch of seeds. Lambda is the density. The second set of parameters is the shape parameters of those inclusions of the seeds. So sigma x, y, and z allows you control the size and the shape of those inclusions. And alpha is the cut level at which we cut those little mountains that we generate by putting the seeds in there. So again, to go through this graphic is basically the skeleton of the method. We create a domain, we throw in the kernels or the inclusions with the density of lambda, and we produce the first Poisson field. And then based on this one, we generate 4 domains. And then we do a weighted geometric average, and we create one more. And then we start we compute the material properties. And then if the material properties match the target, we're done. If they're not, we do new input parameters. We modify these shape factors. We produce new shape, new weights, and we iterate until this is done. So the reason this method works so fast is here. In traditional numeric design generation, you have to generate this field from scratch every single time in the optimization loop. And this is really computationally expensive. So what we do instead, we generate it just once, and then we just iterate here using our shape parameters and the weights with minor adjustments. And this is what creates this huge advantage in computational speed compared to the competitors, so to speak, of this method. So this is the schema. Now, that was the method used to generate the material. Now, how do we compute the material properties? And for that, we use the pore topology method that another student in my group has developed. It is a fast and computationally efficient algorithm. And what it does, as you see on this very simple example of a 3D prism, it extracts the medial surface of the void space. And the medial surface basically is a surface that runs between the solid clusters within the porous space. And then this medial surface has multiple functions. It actually characterizes the void space. So we know what the void space looks like based on the shape of the surface. We can use it to reconstruct the material entirely because it holds all the information that we need about the material. And we also use it as a solution domain for flow simulation. So all simulations, all material calculations happen on this surface. So as an example, here is a 80% porous material. This is the solid phase. This is the void phase. This is the medial surface that goes through the void space. And this is both the solid and the void space. So this is just a demonstration of the concept of the pore topology method and how it represents the material itself. So with these materials that we now can generate numerically, we did several experiments. So in the first experiment, the goal was to match the mean and the standard deviation of pore size at multiple porosities. So we aimed for a mean of 6 micron and the standard deviation of 3 micron, and we generated 20 to 40 domains. And here you see the porosity is ranging from 0.1 to 0.9. The red one is the mean pore size. The blue is the standard deviation. The standard lines are the targets. This is what we want to hit. 6 in pore size and 3 in standard deviation. And this is basically our ensemble calculations. And dashed line is the average. And you see that we can get to very good agreement, especially at the porosities of 0.3, 0.4 and higher. The ensemble of materials matches with the maximum error that is below 11%. So we're pretty happy with that result. Now, for the mean, the error was about 4% and 5% for standard deviation on average. For the second experiment, our objective was to maximize permeability with the fixed mean pore size at 9 different porosities. So here are again porosities from 0.1 to 0.9. The black line is the target permeability that we want to achieve, and the red one is the average of our ensembles, and this red cloud shows 1 standard deviation. We have an excellent match for materials with porosity of greater than 0.2. So from after 0.2, we have very good agreement. The agreement between the target and what we were able to achieve is not as good as at 0.1 and closer to 0.2. And the reason for that is that the optimization scheme solves for shape parameters at maximum permeability. And we have a specific set of kernel locations and kernel orientations. So when you have very small porosity, fixing the kernel locations and orientations means that the connectivity is difficult to optimize. So we're limited in the optimization. But at higher porosities, like on the most of the samples that we were able to generate, the connectivity is not as sensitive to where those kernels end up when we throw them in the domain. So that is the reason why we were able to achieve much better results at porosities of higher than 20%. The third experiment was on computational time. We wanted to know how much better we're doing with adjustable level cut Poisson fields versus its immediate parent, the level cut Poisson field. And we wanted to know what is the effect of domain size or the sample size on computational time. And what we found is that the result, so this is the domain size and this is the time. So the result is pretty similar for small domains, but as the sample size increases, meaning the size of the material sample increases, we achieve dramatically faster results with our modified model as opposed to the original one that we hit. So twice as fast than the original. So we're very happy with these results. So this was all for homogeneous porous materials. Now, our next step was to say, okay, well, can we work with asymmetric materials? Can we generate asymmetric materials with target properties? And in this case, our asymmetric material has a gradient and difference in just one direction. In this case, it's direction Y. And it is different from what I just described for homogeneous materials in 4 important ways. First one, the Poisson field that you see here is not constant throughout the domain. So the density is a function of Y. So this is the Y dimension. So you see much denser kernels here than closer to the bottom. Similarly, other parameters such as the weights that we will give to each one of these domains are different. And the cut level is also a function of Y. So the result is that the material properties such as pore size and porosity become a function of Y, and this is how you control the gradient of pore size and pore distribution along one dimension. So our fourth experiment is we wanted to see if we can produce an asymmetric domain. And here we have showing control of microstructure that produces an approximately linear pore size gradient with the ratio of 5. And the pore size ratio is defined as pore size on one of the domain that is 5 times bigger than the opposite end. So on this axis, we see the kernel density. It's seeds per micrometer square. And on this axis, on the horizontal axis, we see the distance from the top to the bottom of the random field. And we see that the density is much higher at the top of the random field, and then it just decreases towards the bottom. And this is how we can control the pore size of the void space throughout the entire domain. This is what that final domain looks like. You see it is much denser at the top, and it has much more void space at the bottom. And the cross sections also demonstrate that. So we see here this cross section and this one do show this gradient. So the top one shows density of solid phase here and going more towards void at the bottom. Same happens here, more solids at the top than the bottom. But if we cut it in the horizontal plane and show it here, it is about the same distribution of solid inclusions. One more experiment. We wanted to see, can we change the shape of the solid inclusions? So what we have here is fibrous, spherical, and disc-shaped inclusions. And here is the pore size ratio. So when the pore size ratio is 1, it means that the top and the bottom have the same pore sizes. So this really is a homogeneous material. And then from 2 to 10, we are increasing the pore size ratio from 2 to 10. And so you see clearly the differences in microstructure. Here we have fibers, spheres, and discs. Here there is no gradient. And then as you move to the right, you see an increase in the gradient of the pore space. And we have the experimental results here showing cases for fibrous, spherical, and disc-shaped inclusions. On the vertical axis is the pore size ratio. Blue is the target that we want to achieve, and orange is the achieved pore size. And these are our sample numbers. So 1, 7, and 13 are homogeneous, so there was no gradient. And you see that we can hit the pore size very well, no matter what is the type of the inclusion and what is the pore size ratio. So what we find is that constraining input parameters helps us control both the solid structure and the void space, and the error between what we wanted to achieve and what we achieved is under 3.5%. So we're happy with that also. And our sixth and final experiment, we wanted to see how well does the pore topology method perform. That is the one that computes all of these material properties, whether it's porosity, pore sizes, or permeability, compared to direct numerical simulation. And we find that the difference only is under 7% of between direct numerical simulation, our own in-house method. The maximum difference is under 20%. Again, it happens at low porosities where we knew we had some issues. But look at the computational speed. For the pore topology method, we need less than a minute on a personal laptop with this, whereas the equivalent computation with direct numerical simulation is more than an hour on a high performance computer. So if you want really quick iteration of material properties and designing a material, calculating its property, and really fast iterating and finding what is the optimal material that you want, it is clearly advantageous to work with fast numerical methods. So in conclusion, the adjustable level cut Poisson field method is successful in designing porous materials. We showed that we can design homogeneous materials with target properties. And those are pore size distribution, porosity, and permeability. We can also produce asymmetric materials with target microstructures and pore gradient. We are able to control the microstructure and the solid and void space within it. The methods are computationally efficient, and just a small set of parameters of kernel density, shape functions, shape parameters, and the cut level defines the entire space. So these are physically relevant, simple input parameters. And the next step here is to start thinking about how do we go back to synthesis now that we have efficient numerical techniques to generate materials in silico. Now, what is the relationship between the input parameters that we use in our numerical design to actual synthesis parameters? So that will be our next frontier. So that's all for me today. Thank you so much for listening and inviting me. I'm happy to take any questions.
Okay, Lily, thanks very much for the very interesting talk. Now we are going to move to the section of discussion. You can ask questions for any one of the 4 speakers of the section. To do that, you can just unmute yourself and pose the question, or also there is another possibility that you can raise hands. And when you do that, your name goes to the top of the list, and I will pass the mic. I will unmute yourself also, and then you can pose the question. So the forum now is open for questions, whatever. I think there's someone here who wants to make questions. Yes, Rosangela, are you? I'm sorry. Sorry, I thought since you put your video, I thought you were going to ask a question.
May I ask a question?
Sure.
I was a little late to Professor Rosangela's talk, and I know that she was talking about other types of polymer networks, and I thought that I captured some conversation about reactivity to pH. So I was curious if what kind of reaction is that to pH, and is it continuous or discontinuous?
Okay, thank you for your question. In reality, the pH is the catalyst. The cross-link reaction only happens in the alkaline pH. Okay.
I see. Yeah. Got it. Thank you.
Very interesting in your lecture, and I think that you could start a collaboration, because
yes,
I prepare the physical gels and you can make computational simulators.
I think that would be great. I will type my email address. Please contact me. It would be great.
So Lily, okay. I have a couple of questions. Very interesting case, the topological method to choose for the target functions you have. I think you can have multiple solutions now for this target variables that you mentioned. This is a typical problem that happens in any optimization procedure. So then, for instance, if your filter, if your method choose one, you are happy with this one, or you also compare with different solutions?
So
Because
if I understand your question correctly, you also, every time we generate a domain, it's one of the realizations, right? But,
okay.
so-
It's one of the realizations. No?
Yeah.
Okay. So you can have multiple realizations that will fulfill the target properties you have, right?
That's correct. Within one standard deviation, we're still very close. Yeah.
But these realizations, do you postulate a prior PDF for them, or no? They are.
So the only thing that, so the input parameters are the kernel density, which is basically a Poisson field. So you provide the intensity of the Poisson field, and you can say, is it one number throughout the entire domain, 3D domain, or is it a function of where I am in that domain? So if it's not a function of space, then you get homogeneous materials. So every time you produce a material, is just one realization of a Poisson field with that intensity.
Okay, I got it. Okay.
Marcio,
Yes.
I'm sorry.
Go ahead.
Marcio, we have two questions. Anderson.
Okay.
But we have a first question in the chat here. I have a first question.
But there's also an
Jorge Avendanho has a question to Professor Rosangela. I'm the chat, Marcio.
Okay.
Jorge Avendanho. Okay, to Professor Rosangela. I would like to know what the size of the microspheres in thermoresponsive polymer of magnetic chitosan.
They are very the size wise 5 micrometers. There is no no uniformity in the size of the microspheres, but their variety was 5 micrometers.
Okay. Georges, if you have another question, you can ask or write in the chat, please. Anderson, the microphone is with you.
Okay, thank you. I have a question for Lilith. Lilith, maybe I missed something in your talk. I had to respond to a few requests from the kids here, so at some point I disappeared from your talk. But I was wondering if you can incorporate information from petrography, from real materials. I'm thinking about geological materials. If you can incorporate real distributions to populate your simulations at the beginning, so that they would have a more realistic bias in a sense, either from petrological or from NMR or any other kind of tomography.
So the inspiration for the asymmetric materials actually came from membrane work that our colleagues have done at Cornell University. And what they can do is basically they experimentally control the structure of the membrane. It is highly regular and dense at the top, and then at the bottom it becomes more like sponge-like, and it can be it can have round, mostly round porous media, or it can be finger-like. And so that was the inspiration for it. We have analyzed some of their samples, and we try to extract the structure and kind of match it to some degree to the input parameters that we have. And that is work that needs to be continued, because the next stage is really to understand how do you match the synthesis parameters with the numerical entrance parameters. So we use their CT scans to generate the images that we have.
Okay, thank you. Very nice, very neat approach. Thank you.
Thank you, Anderson.
Okay. Again, we have a question from Rodrigo. Rodrigo?
Oh, yes. No,
You have a question also in the chat. The question of the chat I can do after. Rodrigo first.
Yeah, I just received a question from the chat also from Julia Petrova. How do you make a 3D model of the heart? You use the MRI to model the heart of a real person. So yes, the 3D models we generate are based on MRI images. So we have to do the segmentation, the mesh generation, and everything. So we have like 2 years ago we call it patient-specific model. So there was a heart of a particular person, and now they come up with a new name, new cool name. It's the digital twin. So it's a digital twin of your heart. So yeah, we have a patient-specific, and then he asked which programs do you use to make a simulation of the programs we develop ourselves. So mainly using C, or sometimes, so this for the perfusion we use Phoenix, so a Python library which is very easy to use. Sometimes we use finite elements, sometimes we use finite volume, depends on the problem. But thanks for the questions, Julia. I have a couple of questions also to Elcio, if I may. Elcio, you have shown when you showed compare MCMC with Kalman and ABC. What was your target? Was because you said, oh, indirect measuring, targeting. What was your target when you were trying to use these models, these methods to your problem?
Yeah, Rodrigo, thanks for the question. Some examples that I gave, we were interested on the indirect measurement of the temperature, of the transient temperature field inside the whole domain. I was not actually comparing the MCMC and Kalman and the particle filter. These were different examples that we applied. So, for example, for the temperature measurements with the photoacoustic technique, when we wanted one single shot, one single image of the temperature, we used MCMC. But when we wanted like a transient measurements, like at different times in sequence, we have used Kalman.
Oh, okay.
And for the other examples, most of the time we were dealing with non-linear problems, and which Kalman is not suitable to. So we had to apply other techniques, including this particle filter.
Yes. Oh, very nice. And when you said also, when you showed the ABC, that you were the approximate Bayesian computing, yeah? When you were trying to choose the different models, how these models were different from each other?
Yeah, this we apply this in the for the thermal damage, and also for the dynamics of the number of cells in a cell culture, and this algorithm that we have used for ABC, it's capable of both model selection and model calibration.
Okay.
So at the end, you can get the model most suitable to your experimental data and with the parameters most suitable also for that model to represent that data.
Okay.
For example, for the growth of cells, we have applied different models like the logistic model, the extended logistic, and for the thermal damage, these were different models with different complexity levels, like the simple Arrhenius and also models that consider a two-step or a three-step reaction
Okay.
to simulate the thermal damage to the cells.
Okay, thank you very much. It was a very nice presentation.
Thank you, thank you. If I can I ask you also a question?
Yeah, sure.
I mean, I'm
not the chair, so.
I asked the organizer if I can go ahead and ask the question now. Okay.
Yeah, sure.
Yeah.
Okay.
Go ahead.
Go ahead.
Rodrigo,
Go ahead.
couple the electrophysiology model to. Thank you. Couple the electrophysiology model to the model.
I think he lost connection.
Yeah, he lost. We have
lost connection. Okay.
Márcio, we have another question from the chat.
In the chat, yes, I saw.
Yes, we have a question from Dr. Fátima Yubeiro from National University of Asunción, Paraguay. The question is for the Professor Rodrigo.
Lily or Rodrigo?
The question, Rodrigo, the Professor Fátima Yubeiro has a question for you. Is possible to study the relation with pores and donor effect through the membranes?
She was talking about donor effect, but I think she mean osmotic pressure, right?
Yeah, probably.
This is what she call, I think it's osmotic pressure.
Yeah.
I mean, we do study, if it's osmotic pressure, we do study for the perfusion. When we have edema formation, we do include osmosis in our models. Yeah, so yeah, we do include these in the models. Yeah, for the hydrodynamic. Yeah, for the hydrodynamic part. So for the when we model the hemodynamics, so when there is when there's for instance some concentrations of neutrophil or some other particle that is migrating from the intravascular to the extravascular, we do use osmosis for that. Yeah.
Okay.
We have again Elcio.
Yeah.
Is here.
Go ahead, Elcio. You're posing the question, then you lost connection.
Yeah, I'm sorry. I'm sorry.
No, okay.
Rodrigo.
Yes.
Rodrigo, I was asking if you have coupled your model to the electrophysiology model for the heart, like.
Yeah, we made a lot of different couplings. We coupled already the electrics with the contraction, so the heart is so this electromechanics is coupled. We have coupled also this perfusion model with a poroelastic model for the perfusion, with an immune system response model. So we have all the neutrophils and different leukocytes and cytokines, how they answer to a virus or bacterial infection, and how these affect all the pore elasticity, so the tissue dynamics and everything. And where else we have coupled? And we have coupled a little bit the hemodynamics, but only in the arteries, not in the cavities, not with the real, like in the pump, but the hemodynamics.
Yeah, what I mean is, for example, for the heart muscle, have you used like an electrophysiology model for the electric propagation?
Oh yes, we always yes. So we started with electrophysiology. Everything we started with models for electrophysiology. Then we started to couple mechanics, perfusion, hemodynamics. Yeah, but we started, my first models 20 years ago were only for the electrophysiology of the heart.
And which model would you suggest for someone that is starting in this business?
It depends.
The electrophysiology.
It depends
the
on the
canal model or
monodomain. I would say monodomain. The cable model, it's very easy one.
Okay.
And for cell model, then depends on for human, I would suggest 10 Tusher model, which is for human left ventricle. But there are, but a very general and very good model, which is only 4 equations, is from the minimum model from Alfonso Bueno Oroville from Oxford, which is a colleague from mine. It's a very good model for ventricles also. But I would, for the partial differential equation, I would suggest the cable for the monodomain or the cable equation, which is very, very good. Like just a reaction diffusion model.
Okay. Thank you. Thank you.
Oh, well, welcome.
Rodrigo, just one quick question.
Yes.
I saw in your equations that you have the flow, no, the Darcy's law, and then you have three concentrations, right? One you have advection, the other is just diffusion, and the third one is just absorption, right?
Yes. Yes.
Yeah, okay. And then you have also exchange terms, right?
Yes. Yes.
You have these exchange terms. Do you postulate a law for them? It's whatever proportional to the concentration difference.
Yes.
Or okay.
Yes. So, like for instance, that was similar to the question of osmosis. For instance, in the exchange for intravascular to extravascular, we can use Starling law, so that depending on so it's Starling law, it's a mix of osmosis and diffusion. So it brings together osmosis and diffusion mechanisms. So yes, we do have sometimes it can be very simple as a linear model. Sometimes there are more complicated models with non-linear terms. Like normally saturation terms.
Okay.
Yes.
Got it. Okay. Thank you.
Thanks, Márcio.
We have now Yula Petrova.
Yeah. Hello.
Yulia. Okay.
Yeah. A question to Rodrigo. So, did you try to model autoimmune diseases, for example, like multiple sclerosis or the process of myelinization, this stuff?
That was for me. Sorry, I didn't hear.
Yeah. For Rodrigo, yeah.
Okay. Yes. Yeah. Thanks. So for myelinization, yes, we did model myelinization in a particular paper for neuro. Once was published in Phys Review E for myelinization. For autosclerosis, we never model autoimmune. But for the heart, we have model heart and other tissues. We have model many different immune response to different viruses and bacteria, E. coli bacteria, for abscess formation and other kind of diseases. Dengue fever, yellow fever. I have a couple of models for that. But thanks for the question. Yeah.
Okay, thank you.
Thank you, Yulia.
So I have a question to Lily, if I may.
Sure.
So Lily, a nice presentation.
Okay.
When you say you target the permeability, yes. So I thought permeability was more like a continuum parameter that you need to homogenize somehow to get these I want this kind of a permeability tensor. And how do you target a permeability tensor? I got confused with this. Sorry if it's a naive question, a stupid question.
Not at all. Thank you, Rodrigo. So one sample would have one value for permeability. So we want to, let's say, have value X for permeability. So we design the material structure such that the resulting value for permeability will be that number. And we can get very close to it.
And the way you calculate the permeability is how is it? Via homogenization or discrete?
No, this is voxel by voxel, basically.
Voxel by voxel.
Yeah.
Okay.
But then you have to postulate some form of correlation between permeability and porosity.
I think so,
right?
Yeah.
Because permeability is it needs flow simulations. But since you are doing a static version of it, you are somehow exploring some correlations of permeability with the porosity and surface area. Or
so you
like
you have a material with a certain porosity, and within that porosity you can change the structure such that you achieve different permeability. And the calculation is really simple. We use the pore topology method. We basically march on the medial surface, and at every point we we estimate the flow based on the pressure. So it's the very simple calculation, really, for quasi-static.
Oh, so you do estimate the flow, and so to try to extract the permeability.
Yeah, that's right.
That's very interesting. You have papers on that? I would like to read some papers.
Yes, I can send them.
Okay, that's very interesting.
So I didn't get it.
Sorry. Another question is that you mentioned the heterogeneity. Can you also generate a material that is anisotropic? How would you do that?
So the isotropic material is the simplest one because everything is random there. You do not need to impose any gradients.
Yes.
And so again, we have the seed density. So imagine an empty box, and then in the empty box, you throw in randomly some seeds. So that is those inclusions become the foundation for the solid phase. And the inclusions then can be spherical, they can be in shape of fibers or discs or ovals, and you control that using shape parameters, which is delta X, Y, and Z. So it's an ovaloid that you can shrink and contract, make it longer in any direction you want. And then you grow them, and then the final parameter is the cut level. So you grow them to some point, and then you cut, and that cut determines the porosity of the final sample. So for homogeneous materials or isotropic materials, when you throw in those seeds, you throw them in randomly. So you have that Poisson field that is random inside. Now, if you want to create asymmetry, then all of those parameters become a function of space.
Okay.
In my case, it was just along one dimension because I wanted to have dense material at the top and less dense at the bottom. So we created I had a pore size ratio of 5 that I wanted to achieve. And all of the input parameters were a function of height across the membrane, across the material.
These I got, this is for the heterogeneous, but I mean, if you wanted like an anisotropic, like an orthotropic material, like if you wanted to, as a target, a tensor, a permeability, full permeability tensor with 6 different numbers in the tensor, Can you do that?
So this is the case when you move to fibrous materials, right?
Yes,
you mentioned about shape and geometry. When you have spherical, okay, but when you go to fibrous and others, it turns into a tensor. It's not a scalar quantity anymore.
I'm sorry, I thought, Rodrigo, you were saying isotropic, so that's what
I'm isotropic, sorry.
Yeah,
no,
anisotropic, yes. Yes.
Anisotropic, yeah,
Yes.
autotropic or something like
yes,
that.
Yes, you basically restrict your input parameters in whichever way you want, depending on which direction you want to control.
Okay.
Very nice, very nice. Very
Okay.
interesting.
So then you mentioned that you solved the flow problem also. You mentioned about pressure. You mentioned about pressure and flow. Do you solve the flow problem also to have an estimate of the permeability?
Yes, yeah.
Oh, okay.
So the first method is you create the material, and the second one is when you calculate the properties. So that's pore topology method. So we take the numerical sample, we extract the pore medial surface of the void space, and then we use that medial surface as the solution domain. So we march voxel by voxel when we compute local hydraulic conductivity at each voxel.
But how do you calculate this local permeability within each voxel? Do you use some sort of correlation, or
No,
how do you have information of
so I have information about the viscosity, and I have information about the distance to the solid walls. So I assume parallel flow at any point in on the surface.
Okay, so use some sort of Poiseuille law or something like that. Okay. So this is the correlation which I was asking.
That's very interesting.
I
wonder if this matches a homogenization method.
So the domains that we looked at are maximum 250 voxels by 250. So I think we are hitting the REV, but we did not try to compare head to head with any other methods. We just did the pore scale simulations.
You mentioned also that it was very fast, but I did some fast calculations and it was something like 6 hours. Yeah, is this in a single computer? Because it was 10 to the 4
under a minute.
seconds?
Under a minute.
Oh.
Under a minute on a laptop.
I thought it was like some hours.
No.
10 to the 4.
So we compared these methods with high performance computer for direct numerical simulation. So where we are going under a minute, there is more than an hour to compute with direct numerical simulation if you want to do that. So our calculations are very fast.
That's very nice. Okay, thanks. And they are like, so you're not using any GPU or parallelization yet?
No,
just
no, laptop, just old laptop.
Cool. Yeah, it's nice.
Very fast.
Thank you.
And Lily, one more thing. You've mentioned about the hydrogel, right? And then your target functions are functions for the flow problem, porosity. I think that for the hydrogel, you also need some sort of target for the swelling property. Can you do extensions for this to have a better, like some sort of target of swelling property? Or I don't know if you can do the same thing for the charge density. You can build up these polymers with a certain electrical charge, right? And can you also include these in the target functions?
I hope so. So we have not done that yet, but definitely we want to include the elasticity and the deformation because that's a big part of it. So the reason I was focused on the flow is the speed of the sensor depends on how much it will increase in volume and decrease in volume and how fast. And that basically a flow problem. How quickly the liquid will leave the polymer network and how quickly it will get into the polymer network. That's why we wanted to optimize for permeability. We wanted to optimize for just the microstructure and understand the relationship between the microstructure and the speed with which it can shrink and expand. So, but the calculations that I showed you do not include that swelling behavior, and that would be the next step. So ideally, you would have the same, if you think about it, if you don't have serious deformations, then your medial surface will be the same. It's just the distance between the center of the medial spaces and the void space and the nearest solid phase is going to change as the hydrogel expands. So you would have faster flow, but that is something that we need to test. We haven't done that yet.
And the surface charge, it's also, you can also put in terms of a target function, or it's fixed the surface charge density of the polymer? It's
that probably will become one of the attributes at each voxel of the medial surface, because the medial surface is something that we use that, it really contains all of the information about the entire sample, both the void space and the solid space. So what we want to also add is the contact angle should be specified at each voxel, ideally, if you really want to be very careful about how you model the flow. And if we can add the electrical charge or any other chemical properties, that's where I would target it as attributes at the surface, at the medial surface.
I see.
Because if you want to reconstruct the entire material, you want to have a repository of all the information about it, and the medial surface is that repository.
Okay. Is there any other question, Siddhartha?
No question.
So I have one for Elcio.
Okay.
Elcio, in your steps of your, whatever your method, the Kalman filter or the Markov chain Monte Carlo, do you postulate some sort of prior for the thermal conductivities of for the coefficients of the bioheat equation, right? You need some input there, which is the thermal conductivities and the exchange parameter. Does these parameters are up? Their values change when you are using whatever Kalman or Markov chain? Do you postulate a prior for them, and you have a posteriori also? I didn't get this.
Yeah, it's a very good question, Marcio. And for example, for Markov chain Monte Carlo, you specify the priors, and actually all the model parameters, they are considered as random quantities. So for all parameters, you generate a posterior based on the prior information that you have assigned, and also the input that you get from your measurements. As for the Kalman or for the particle filter, we also specify priors, and with those priors, we make Monte Carlo simulations to get the uncertainties for the model as a global. So when we simulate those uncertainties as an additive error, and we try to use additive and Gaussian uncertainties for the evolution model. But these uncertainties, they are modeled based on the prior information that we have for the parameters that are propagated for the responses, for the dependent variables based on some Monte Carlo simulations.
Yeah, and then you have one thermal conductivity for the tumor and another one for the healthy tissue.
Yes, usually each tissue layer has a value of thermal conductivity and blood perfusion based on the values that we can get in the literature. So we did for different tissues. Of my students, he did a literature search to get like all the values that have been reported. And so based on these values, we have like means and standard deviations, and also lower and upper bounds. And most of the time we have been using truncated Gaussian distribution based on these means and standard deviation, the minimum and maximum values. So we make sure that we are not out of those ranges that have been reported.
Okay. Thanks.
Okay.
Thank you.
Another question? If you have a question, you can use the chat also. If you have a problem with the microphone, you can write in the chat, please. Another question? Professor Rosangela, I think that Lilith write the email here in the chat. We can find the chat. Did you see the email?
Yes, yes.
Okay, great.
Thank you.
So
More questions?
No more questions.
No more questions, Monsieur.
Okay. So we'd like to thank all of the speakers and the audience. Thanks very much for accepting our invitation. And we are going to have 2 more sections tomorrow. One in oil and gas, and the second one in microfluidic. So I hope to see you there. And thanks very much, Lily and Elcio, Rodrigo, and Rosangela for giving the talks. I don't know if Siddhartha wants to say anything more.
Okay, just thanks all the accepting professor, all the talks, and I'd like to find everybody tomorrow.
Just to remind. No, thank you very much to everyone. And just to remind that the link is different. So we have 4 different links for the talks. Okay, thank you very much, everyone. It was really inspiring.
Thank you very much. Hope to see everybody here tomorrow. Okay. Thank you.
Bye-bye.
Thank you.
Bye-bye. Until tomorrow. Bye. Nice to see you all.
It was a pleasure to be here.